Dr. Jaffe, this is regarding the LRA by Lysac testing. Can you explain how the activation of kinase enzymes on the glycocalyx membrane of a lymphocyte indicates lymphocytic activation? And how does it show us one versus, all three types of delayed lymphocytic reactions?
Um, this goes to the fundamental principle on which the test relies. When we started, we knew that when an antigen-presenting cell wiggles over to a lymphocyte and presents at something called the MHC, Major Histocompatibility Center locus, a processed antigen that the cell has already been programmed to recognize, like a lock and a key. What we knew was that phosphate was very involved. We knew that phosphate went and was added by some enzyme. We had an idea that turned out to be accurate, to be revealed. But we knew that when that activation of the cell occurs, the very first step is to phosphorylate the glycoproteins, the glycocalyx, the hair on the cell. And phosphate is one of the strongest electrical charge molecules in biology. So the more phosphate you put on these glycocalyx proteins, the more they spread out and push away the plasma and everything else, creating what looks visually in the microscope looks like a halo. But the halo is the absence of plasma, and the absence of plasma is due to the phosphorylation of these glycoproteins. Now the same process, the absolutely same process, sends other phosphorylated molecules to the nucleus and tells the nucleus to go into mitosis and cell division. And if you wait 3 weeks, you might get a colony. If you wait 3 days with the right radioisotope, you might see thymidine incorporated into DNA. But I knew that we did not have that much time. I knew that radioisotopes were getting more and more problematic to work with. And I'm a chemist, biochemist by training. So I said, phosphate kinase. And in a 1987 now classic draft article, I think I even mentioned JAK2, which it turns out to be. There's many in this family called JAK kinase family. They send lots of molecules, messenger molecules, powerful messenger molecules. And we were fortunate because we started with the premise that we were going to break the mold and we were going to do ex vivo cell culture in the laboratory even 3 days after a specimen was drawn. So we had to be accurate about the method in some detail to preserve the viability of the cells so that in the laboratory they could react just as they do in the body. And we had to pioneer a number of aspects of what we do and have done and continue to do, but very precisely. So our split sample data is the best in the cell culture world and rivals any laboratory anywhere. If you're looking for perfection, don't even do an analysis. Nothing is perfect, but we are well above excellent and been tested over many years clinically, because we have an interpretation that says, what do you do about this? How do you benefit from the challenge? How do you relieve the affliction and restore digestion and the communication between the microbiome and the gut nervous system, and then the gut nervous system talking to the central nervous system, which is now talking to the whole body? So it's all connected from a systems point of view. And we were fortunate enough to figure out what it meant that these pairs, these glycoproteins, stood up. Okay? Others showed that this happened. We figured out what it meant for lymphocytes. And now, since you also asked kindly, how do you know it's a type 2, 3, or 4 reaction? I can tell you how we could know. I can tell you what the additional laboratory procedure, effort, resources, and so forth to differentiate are. But what I can tell you, and this is the clinically important piece of information, if it's a helpful neutralizing antibody of the type 2 kind, ignore it. That means any physical chemistry that measures an antibody without knowing whether it's helpful or harmful is now passé. But if it's a harmful B-class antibody producing lymphocyte, I want to know about that. And what if it's an immune complex? What if it's IgM, anti-IgG antigen, and this is the body's effort to protect itself? Think of things like temporal arteritis and glomerulonephritis. If it's a type 3 reaction, I want to know it. And then what about those little T cells that react, they're lymphocytes, that react without antibodies? Oh, I absolutely need to know about that, because that's where autoimmunity or tolerance, intolerance or tolerance, largely resides with the type 4 T cells. However, don't forget about the immune complexes and the harmful B cell reactions. The complement fixing B cell, the cytolytic B cell antibodies, as opposed to the neutralizing antibodies, which are the super majority, which say, in essence, doesn't matter. So we measure clinically all three reactions concurrently, and what we measure is lymphocyte activation, and specifically lymphocytes, not just particles, not just anything that's a certain number of microns, lymphocytes. Now, we could, with much more elegant procedures and lots more resources, actually confirm which were the type 2 harmful B, which were the type 3 immune complexes, which are the T cell reactions. We don't do that on an individual basis. It's a fair question. And I'm saying clinically, you need to know helpful from harmful. And you need to know that just as it happens in the body. So that's why we identified this first step in lymphocyte activation that almost immediately, this is an enzyme, it's a catalyst, phosphorylates the glycocalyx and pushes the plasma away. And come take a look sometime. We welcome people to schedule a time in the lab. And we'll show you how careful we are and why it matters. Because we have the most cell cultures done, but the most cell cultures done accurately with blind split samples spread over many years, reported at the American Society for Investigative Pathology, and other similar proper and rigorous internal and external controls. So put us to the test. Let us help you restore the microbiome and digestion based on what you eat and drink, but include what you think and do. And we weave all of that together in the interpretation of each test.